Really great honor and pleasure to introduce Paul Bieniasz tonight, who's our keynote speaker, as you know. So Paul doesn't really need any introduction, of course, but I'd like to give him one anyway, if you'll bear with me for 2 minutes. Paul got his PhD studying virology at Imperial College, University of London, in 1996. And then he came to the United States and joined Brian Cullen's lab at Duke University, where he worked on various aspects of HIV until about 1999, when he moved to where he is now currently, which is the Rockefeller University and Aaron Diamond Research Center. So he is now a professor, head of the lab of retrovirology, and an investigator of the Howard Hughes Medical Institute. Well, you know, in keeping with the tradition of terminology at this meeting, I would like to say that Paul, in the last 15 years, has really found out a bunch of really cool things. But we've heard from at least 3 speakers that bunch is not a very technical word, and cool things probably isn't either. So more formally, I would like to say that the breadth and depth of Paul's research achievements is really remarkable. And his interests, as most of us know, or probably all of us know, have included retroviral assembly, where he made important contributions to the identification of ESCRT proteins in the retroviral budding process. He's been interested in aspects of retroviral evolution, and that included the resurrection and characterization of ancient retroviral sequences. And of course, he's put a large amount of his efforts into understanding intrinsic antiviral immunity, what we call in this field restriction factors, although we seem to have some trouble knowing exactly what those are. So I would say that through these accomplishments, Paul has established himself as a leader in retrovirology. And beyond this, and moreover, he's been a very generous colleague and collaborator, and I think we can see that just from the meeting where we hear his name and we see his name in the acknowledgement slides. And so he's made tremendous contributions to this field. So I would like to say that we're all very lucky to have Paul as a member of this research community and to have him as a colleague. So for my own sake, or it's not my own sake, from my own point of view, I would like to say that every time I have a chance to chat with Paul about things retroviral or otherwise, I'm reminded of how much fun and how much I enjoy and value his insights and perspectives. So I'm really looking forward to this next hour, as I'm sure you are, because I'm very confident that we are going to hear a bunch of really cool things. And so, Paul, thanks for accepting my invitation.
Well, thank you very much, John, for that very generous introduction. Thank you and welcome for the invitation to be here. Although I have to say it was much more of a pleasure to be asked to give this talk than to actually give it. Because it's actually quite a difficult talk to give. Various people over the years have approached this talk with various different recipes, but what I'm going to do is tell you a little bit about myself, at risk of sounding vain, but I'll try not to be. Then I will give you a few highlights of the work that my lab has done, and that'll serve as a background for a little bit of new stuff that I'll introduce into the talk. So this is me around 1995. That wasn't supposed to be funny. Maybe you were laughing at Myra. No, I worked in Myra McClure's lab, who is an extremely generous and nurturing mentor. You might recognize her for her more recent notoriety in helping to at least put the first dent in the XMRV CFS hypothesis. But Myra had an incredibly wide range of interests, which influenced me to this day. And so when I worked with her, I worked on HIV, but my thesis was actually on one of the underrepresented minority retroviruses, the foamy-viruses, and I did lots of different things with those viruses. Really was a very good training ground for graduate students. Competition was a little less intense than in HIV. One of the best things that Myra did for me was to send me to this meeting. And so I first came to this meeting in 1995 and was absolutely blown away by the breadth and depth and excitement of the science that I witnessed here. I met someone who was attending this meeting for the first time the other day, and they had a particularly apt analogy. They said it's like trying to drink water from a fire hydrant. And I thought that really mirrored my own experience. The other thing that was quite important for me at this meeting is that I met my future postdoc mentor, who was Brian Cullen. He had a bit of a fearsome reputation at the time, so I was a little reticent about approaching him, but he actually turned out to be an incredibly generous, warm, wonderful mentor who's been supportive of me throughout my career, both during the time I was in his lab working on these things and since. I also gave my first talk at Cold Spring Harbor when I was in his lab, which was one of the most stressful things I've ever done. But also when I was in his lab, I thought I'd try my hand at an alternative career. And you can see I was very enthusiastic, rather a little too enthusiastic, but my obvious enthusiasm wasn't matched by obvious talent at playing the guitar. So I hung the guitar up and found a proper job when I finished with Brian. So I moved to New York around the end of 1999 to start my laboratory, and I didn't have a clue what I was doing, but based on some of the work that I'd done in Brian's lab, I thought a good idea would be to try and get HIV to replicate in cells of a species that it couldn't replicate in. And over in the previous few years, work from our group and from others had defined what you needed to do in order to make a mouse cell susceptible to HIV entry and able to transcribe the viral genome. So this was the first paper I published from my lab. What I lacked in wisdom, I'd made up for in industry, and so we submitted this paper about 6 months after I started my lab. It was rather a descriptive paper, but I'm quite proud of it. It was a reasonably important piece of work. Ned Landau and Vineet Kawaramani did similar work around the same time. But it was much more important to me personally than it was to the field, because it really set the directions of my lab going forward. And so I suppose it was obvious to many people at the time that it's actually very interesting to study retrovirus replication in cells where it doesn't work. And so restriction of retrovirus tropism became a major theme of my laboratory. And because we thought, maybe erroneously, that there was blocked assembly in rodent cells, then we started to work on HIV particle assembly. And so for the rest of the talk, I'll tell you one or two stories in both of these areas, some old stuff and some more recent stuff. I was also quite strongly influenced by another species-dependent block in HIV replication that I first heard about at this meeting, but was later published in Journal of Virology by Joseph Sadrowski's group. It was undefined at this point, but a post-entry block to HIV replication in primate cells. And so one of the first things, or one of the early things we did, and by this time I had been joined by my then girlfriend and future wife, Theodora Hatziana, was to try and understand the nature of this block to lentivirus replication in primate cells. And so this is one of the first experiments that Theodora did, and what she did was to fuse HIV-susceptible human cells to HIV-resistant monkey cells and show that the heterokaryons were resistant to HIV-1 infection, suggesting the presence of a dominant inhibitor. We also did experiments that showed that if you titrated HIV-1 on resistant monkey cells, then you got these strange multi-hit kinetics, and you could enhance HIV infection in those monkey cells by treating them with VLPs, and all you needed to have in those VLPs was really processed Gag protein. These experiments also showed that capsid was the determinant of this differential tropism, and altogether suggested the existence of restriction factors targeting the HIV-1 capsid. Ned Landau and Greg Towers had made similar observations at around the same time. And Greg actually happened to be in New York, and we, at that time, on a sabbatical in Steve Goff's lab, and so we teamed up over the next year or so to show that the restriction that he and Jonathan Stoy had shown existed for entropic MLV, and they had termed Ref1, was very likely mediated by the same factors that we had shown probably existed and restricted primate lentivirus infection. And we do things like saturate cells with an HIV particle and show that that increased susceptibility to entropic MLV, strongly suggesting the existence of a species variable restriction factor that targeted incoming HIV-1 capsids. We also showed that in some strange cells from owl monkeys, you could apparently completely reverse the restriction with this drug cyclosporin A or mutations in the cyclophilin binding loop. And we had lots of fun working with this phenotype. We invested a lot of physical energy and emotional energy trying to identify what the factor was, and that led to the first major disappointment in my career. And that was when rumors began to circulate that Joe Sadrowski's lab had identified said factor, which we all know now as TRIM5. And so one day, John Cohen, who was writing a piece for Science magazine about the discovery of this factor, called me up because apparently he wanted some other people in the field to give their opinion about the importance of this factor. And I was quite flattered that he would seek my opinion, but I hadn't yet learned really how to deal with journalists, and the idea that you could go on and off the record. I knew that, but it didn't occur to me that I needed to be very clear about that. So instead of a considered intellectual comment about the importance of this work, I said the following: I'm sick as a dog. And this appeared in Science. He at least allowed me to recover some dignity by paying the work a compliment, but I learned a valuable lesson. And thank you to all of those of you who sent me emails poking fun at me about this. Helped me learn a valuable lesson. Anyway, so following that disappointment, we changed tack a little bit, although we continued to work on TRIM5 and published some nice work in that area. What Theodora and I saw from this was an opportunity, perhaps, to do something practical and useful with this discovery. And so we reasoned that it might be reasonably straightforward to change the tropism of HIV-1 from what's a strictly human or human and chimpanzee tropic virus to one that might be able to infect an animal species that you could acquire in some numbers in primate centers, i.e. to make a simian tropic HIV-1. So our first crack at this was basically to change the known tropism determinants that conferred restriction in monkey cells, of course. So we changed the capsid. And of course, Michael Malim's group, even well before they discovered ApoBec3G, had shown that VIF proteins worked in a species-specific way. And so we reasoned that if we change capsid and VIF, we might be able to get HIV-1 to grow in monkey cells. And so this is the result of that experiment. The initial construct was a little bit sick, but with training, it became fitter. And unlike the wild-type HIV-1, which is essentially dead on arrival in rhesus monkey lymphocytes, our engineered virus could replicate very nicely in these rhesus monkey cells, almost as well as SIV-MAC 239. Unfortunately, when we took this into animal experiments, and we did so then, and for all the animal experiments I'll talk to you subsequently about, with a very great team of collaborators, Vineet Kaur-Ramani, Jeff Lifson, Theodora, and myself. Unfortunately, this virus really didn't perform that well in intact animals. We surmised because of the fitness defects that had been introduced by these manipulations were just too great to overcome. Although the possibility exists, there are other blocks. But we got a fortunate turn of events in the discovery made by us and by several other groups, including Welkin and Greg and Shulock Hu and another group whose name I forget, that there's a sort of a natural pseudo-knockout allele of TRIM5, at least with respect to HIV restricting activity in macaques. And so very closely following the discovery of the TRIM5 alpha protein, Jeremy Lubin and then Jonathan Stoy had found that in owl monkeys, a cyclophilin A cDNA had retrotransposed into the TRIM5 locus. I'll never forget the almost audible gasp from the audience when this was announced at this meeting. Converting TRIM5 alpha into a TRIM-CYP protein. Now, a similar but distinct transposition event has occurred in macaques, giving expression of a TRIM-CYP protein. The key difference in our case was, of course, that the cyclophilin A portion of this protein had evolved to chase some other retrovirus and had actually lost the ability to recognize HIV-1, rendering pigtail macaques in which 100% of the TRIM5 alleles are of this type, effectively denuding them of a key HIV-1 restriction factor. And so we were able to take a virus like this without changing capsid, just changing the VIF, and it could grow very nicely in pigtail macaque lymphocytes in vitro. And so in collaboration with Vineet and Jeff's groups, we introduced this virus into pigtail macaques. And here you can see viral load and CD4 counts into the initially inoculated macaques, and you could see we got a reasonable take. The levels of acute viremia are sort of about 1 to 10% of those that you would hope for and would likely observe in an HIV-1 infected human. And the monkeys would either partially or completely control infection, but this at least got our foot in the door. And then ensued an experiment that lasted the best part of 4 years, where what we would do is CD8 deplete this initial macaque and then passage the virus to subsequent generations of macaques. And we've done this over a number of passages. I've neglected to mention that the virus that we're using here is essentially HIV-1 with an SIV MAC-VIF, and it had a cocktail of sort of prototype R5 tropic envelope genes. But over time, the virus clearly heated up. For subsequent passages, what we would do would CD8 deplete the animals at the time of infection. It's important to note what the effect of this CD8 depletion is. It effectively removes CD8 cells from the blood and from the lymph nodes, but doesn't touch CD8 cells in the gut, where about half of them reside. So this is a partial CD8 depletion, and it lasts for the first few weeks. So it's a temporary and partial removal of CD8 cells. And by the time we got to passage 4, 1 of 3 animals succumbed to AIDS with very high viral loads, but from passage 5 and onwards, this has been a reproducible phenotype where if you do this transient CD8 depletion, the monkeys essentially behave like rapid progressors. They have extremely high viral loads and succumb with AIDS-defining conditions or conditions that are frequently found in human AIDS patients. The somewhat interesting thing is that if you do not include the anti-CD8 antibody with the inoculum, the animals behave in a manner that you rather akin to how we understand elite controllers to handle the virus. So you get a nice peak of virus, but then it's rapidly brought under control and remains at relatively low levels thereafter. If you CD8 deplete the animals after infection is established, you get a peak of viremia, but then that's rapidly controlled. So CD8 depletion after infection is established in no way recapitulates the effect if the depletion is applied during infection, tending to suggest that there's something rather critical that goes on, at least in this model, and perhaps more generally, during those first critical few weeks of HIV-1 infection that's essentially deterministic for the clinical outcome of the infection. Over here you can see the CD4 counts associated with these two animals, and they almost invariably drop to extremely low levels as the animals develop clinical AIDS. So obviously an interesting thing to ask is what happened during this animal passage. How did the virus evolve? And we've done that, and I'll tell you a couple of stories related to that as we go on. This is a family tree of envelope sequences that are recovered from the passage series. First thing to note is that although we included 5 different R5 tropic viruses in the inoculum, we might as well not have bothered with 4 of them, because one dominated the population beginning with the first monkey, and that was the 88 strain, which seems actually quite, not uniquely, but there have been other examples where this envelope has been successfully used in monkeys. Other than that, the level of diversity that one sees in these macaques is somewhat unremarkable. It's a slightly apples to oranges comparison, but the amount of divergence in this approximately 3.5-year experiment is roughly the same as you would expect to see in a single infected human for the same, over the same period of time. One thing that did happen in one of the 4 animals that I showed you was a clear example of a co-receptor switch as a result of a small deletion in the V3 loop. This rarely, almost never happens in SIV infected animals, but as you know, happens with reasonable frequency in HIV-1 infected humans. And so a sort of a common feature of HIV-1 infected humans is recapitulated in this model. So to tell you something else that's quite interesting about how the virus evolved in this passage series, I need to digress for a minute to tell you about another line of investigation that happened in the lab. And that was the discovery of tethering made by Stuart Neal. And when Stuart joined the lab, we knew a reasonable amount about VPU function. Work from Klaus Strabel, Heinrich Gottlinger, Paul Spearman and others had established that VPU was required for viral release in a range of cell lines, and the phenotype associated with it was accumulation of virions in intracellular compartments and on cell surfaces. But what wasn't known is what was how did this block arise? Was it a mistargeting effect or anything in very much detail about how the block arose? What Stuart did initially was basically to put some directionality on the system and show that if he blocked endocytosis, then virions accumulated on the surface but not within endosomes. And he was also able to show that the virions could be recovered from the cell surface in mature form with a viral protease, suggesting that assembly had gone to completion, but there was something on the cell surface that was keeping virions stuck there. So Stuart tried a whole bunch of experiments to try and identify this factor. But we were talking about this one day and remembered that there was a literature about a protein called ICAM1 that was known to exist on the surface of cells and increase adherence of virions to cells. And of course, we thought by this time that something like an adhesion molecule might be what's responsible for keeping virions on cells. So Stuart excitedly decided to test this hypothesis that ICAM1 might be the said factor. And Stuart was never the sort of person who would do an experiment tomorrow if he could do it today. And so what he did was to make ICAM1 clones, and as soon as he got the mini-preps, transfected the cells and got a beautiful VPU-dependent virus release. We almost cracked open the champagne at that point, but thought we'd better repeat it, which Stuart did. And this time he did it with some DNA that he'd spent a little more time purifying. And of course, the phenotype completely disappeared. So that was obviously potentially the second major disappointment of my career. But it made us think about things like interferon, what's in dirty DNA, things that might trigger an innate immune response in the cells that one was transfecting. And so that gave us a critical handle, and what we then found was that the requirement for VPU could be easily induced in a range of cell lines just by treating them with interferon. That gave us a handle to go looking for genes that were interferon-induced, expressed at the cell surface, and differentially expressed in cell lines that did or did not have the constitutive VPU requirement. And so this was the first microarray experiment my lab had ever done. And within an hour of getting this data, using very sophisticated analytical software, Microsoft Excel, we plotted a graph of gene expression in response to interferon versus expression in these two cell lines. And we had a candidate. Basically, there was one candidate that was particularly compelling. And the rest of the story was basically like falling off a log to prove that this protein was, in fact, what imposed the requirement for VPU. And you can see here things that I'm sure you've seen many times before, that if you have a VPU-deleted virus and you express tetherin, you get this impressive recapitulation of the virus release defect that had been defined by others. This was a particularly satisfying time in my career, although I wasn't actually in the lab for this discovery because I was on paternity leave, where I used my biological training in the initial stages of fatherhood. This is my son, Stefan, who you'll see in a little less compromising position later. But you can see the euphoria lasted for some time, because at the subsequent Cold Spring Harbor Retroviruses meeting, here's John, who, as you know, independently connected the dots to make the same discovery, and we're sharing a beer here. John's obviously keeping his composure slightly better than I am. But the reason for this digression, in addition to tell you about the story of which I'm quite proud, is to go back to how the virus changed during adaptation in pigtail macaques. And so Matthew McNatt in the lab had, and other labs found the same data, showed that VPU worked in a quite species-specific manner. And so while it was a very effective antagonist of human tetherin, it was basically inactive against tetherin proteins from monkeys, and in particular for this story from the tetherin protein from pigtail macaques. But one of the things that had happened during the passage of HIV-1 in pigtail macaques was effectively a gain of function mutation. There were changes in the transmembrane domain, and Matthew had showed that, and others had showed that the transmembrane domain was really the determinant that governed the sensitivity or otherwise of tetherin proteins to VPUs. There was a gain of function mutation that allowed VPU to now become an effective antagonist of pigtail macaque tetherin. So here's the wild-type VPU doing no better than, the wild-type VPU doing no better than the VPU-deleted virus, but the mutations that arose conferred significant ability to confer resistance to the pigtail macaque tetherin. The other change that we've more recently found is a set of mutations in the capsid protein of the virus as it's been passed through pigtail macaques. I'm rather reticent about bringing up another set of capsid mutants that confer resistance to MX2 proteins after last night's session. But we did notice that there were mutations that arose in the capsid that were either extremely rare or absent in HIV-1 sequences that were found in humans. These are the positions that are occupied by those mutations. They're not all present in every sequence that we get. There are various combinations. Many of them are not fixed, and some of them are. But what's interesting about them is that they appear to confer partial resistance, but specific resistance to the pigtail macaque MX2 and not the human, basically changing it from a 10-fold inhibitor to about a 3-fold inhibitor. Most of these don't have a fitness cost, with the exception of this one, which has a high-level resistance to both MX2 proteins. And while I'd certainly stop short of claiming that MX2 was the driver of these mutations, there are obviously other reasons why those mutations might occur. It's clearly an intriguing possibility that MX2 has exerted some selection pressure on the virus in macaques. So I'm going to leave this section now, just to summarize what I've told you about restriction factors and primate lentivirus tropism. I hope I've convinced you that by selecting the right host, by doing a little bit of viral engineering and adaptation, with the primary goal of avoiding restriction factors, we're able to switch the tropism of HIV-1 in a species that it would not normally infect to one in which it can cause AIDS. The disease that we find in these macaques, I haven't shown you this, but it quite accurately recapitulates key features of AIDS in humans, including occasional receptor switching. And I also showed you this rather interesting phenomenon that very stark differences in outcome of this infection can be determined by manipulations that are confined to the first few weeks of infection. So let me switch now to the other main theme that my laboratory has worked on, and that is HIV assembly. And so, while, as I mentioned, the motivation to work in this field was based on a finding that assembly seemed to be rather poor in murine cells, we've never actually managed to figure out the blocks to HIV-1 replication in that species. But that did inspire a more general interest in the field of particle assembly. And so one of the first things we did was to investigate the role of the ESCRT proteins in viral budding. And this was inspired initially by a paper from Carol Carter's group that had showed that the PTAP motif, which was long known from the work of Heinrich Gottlinger, Eric Fried, and others, that when mutated it would induce this rather interesting defect in virus particle assembly. And so Carol Carter's group had first shown that the PTAP motif could bind to this cellular protein called TSG101. And so what we were then able to show was that you could actually functionally replace the PTAP motif with a TSG101 protein. We set up this transcomplementation assay so that if you took a late domain defective HIV-1 mutant, you could complement it in trans by expressing a Gag protein that also lacked late domains, but was instead fused to TSG101, and that's the key result here. We also showed in this initial paper that viral proteins, we didn't show HIV-1 Gag, that came later, but we used another protein, the Ebola virus VP40 protein, which also had a PTAP motif, could redistribute TSG101 from sites where it was normally found to the plasma membrane. And that didn't happen when you made mutants in the PTAP motif. Now, this might be the worst immunofluorescence picture that my lab has ever published. But I think you'll forgive me when you find out what was going on when I took this. So I spent a day in the microscope room acquiring images like this and others, similarly ropey, because I'd just returned from the roof of our building where I had watched this happen. So this was, just to give you a little bit more background, this was at a very sort of intense period in the lab. We had heard rumors that Wes Sundquist's now classic paper was on the way out, on the way to the journal, and that the window of opportunity for us to make a contribution in this field was rapidly closing. So faced with the choice of going to the microscope room or going home and fretting, I spent the day in the microscope room trying to salvage this set of experiments while the world apparently was literally collapsing around us. This was a uniquely stressful day, as I'm sure you can imagine. But things turned out okay. We managed to get a reasonable publication out of this, and New York City recovered, at least to an extent. And we and others went on to define a whole set of protein interactions. I can't possibly do justice to everyone who contributed to this area. But the bottom line was from studies from a number of groups that we could define a set of proteins that interacted either directly or as a consequence of ubiquitination with Gag proteins or something close to Gag proteins. And these proteins, as components of the ESCRT pathway, their main job was basically to recruit proteins that others have gone on to show are really the key drivers of membrane scission that enable particle release. One of the key points is that these components of the pathway are generally required in a late domain or virus-specific way for virus budding, while this component of the pathway is required in a general sense for particle assembly. So one of the other things that we thought we did get interested in was actually visualizing the process of virion assembly. It was very nice to understand how all these proteins interacted together, but actually seeing them in action was one thing that we were very interested in doing. And one of my colleagues at Rockefeller University is a pioneer, a leader in the implementation of a microscopic technique called total internal reflection microscopy. And when I saw what he could see at the cell surface in terms of watching things endocytose and exocytose, I realized more or less straight away that this would be a uniquely powerful technique in order that we could apply to watch HIV-1 assembly. And so a postdoc in my lab, Nolwenn Juvenet, undertook this project to try and visualize the assembly of individual HIV particles in real time. Bear in mind, the assembly of no virus had been imaged at this time. And I don't think I'll ever tire of watching this movie, because it actually took a long time to get the system to work. But once it did, it did so in a rather aesthetic way. So this is a HeLa cell expressing a mixture of HIV-1 Gag and Gag GFP. And you can see the individual particles gradually appearing at the cell surface over a period of about 40 minutes. And so we could look at these particles more closely. You'll see a movie here where we've looked more closely, and we could quantify the fluorescence associated with these particles over time, and it would follow this sort of stereotypic pattern where there'd be low levels of fluorescence, it would ramp up and plateau, exactly the signature that one would expect of an assembling particle. And Nolwenn, Nolwenn did a whole set of experiments that really cemented the view of these appearing puncta as assembling HIV-1 particles. So we went on to extend this technique in a variety of ways. So one of the things we did was to make fluorescent fusions of the ESCRT proteins, and we could show using EIAV that one of the proteins that was required for its budding, ALEX, accumulated along with Gag and plateaued with it during virion assembly. And in contrast, proteins that acted later in the ESCRT pathway, particularly ESCRT3 and VPS4, only appeared transiently at the end of particle assembly and then were removed from sites of particle budding. We're also able to start imaging other components of the virus, in particular the viral RNA. And so this is a stylized composite of many different types of experiment. We can't possibly watch all of these things at once. But we were able to image individual RNAs or pairs of RNAs that would appear quickly at the plasma membrane and would remain there, and at some time period thereafter, generally within a few minutes, Gag would start to assemble at the site of an RNA molecule and then go on to complete its assembly. And so we were able to derive measurements of particle assembly by analyzing individual virions that would essentially impossible or nearly impossible to get by any other technique. And so this was a very sort of rewarding endeavor. Now, imaging was great, and it told us a lot of the basic parameters of HIV-1 assembly, but doesn't give you sort of molecular details of how individual things interact. And more recently, what we've become interested in is how the viral genome behaves and how it interacts with Gag and with other proteins as it is made in the cytoplasm and is incorporated and assembled into viral particles. And so the field of RNA-protein interactions has actually been quite revolutionized recently by the application of a new set of techniques called CLIP, or cross-linking immunoprecipitation, actually developed by my colleagues at Rockefeller University, Bob Darnell and Tom Tuschl. And what they have done is to basically enable one to define rather precisely the RNA target sites of RNA binding proteins using a cross-linking approach. And so this is a direct binding assay. You really need atomic distances in order to get an efficient cross-link. The assay is performed initially under physiological conditions, and I mean by that live cells. As I'll explain, it gives near nucleotide resolution. It's important to understand the limitations, though. It doesn't give you any assessment of dynamics, affinity, or location unless you couple it to fractionation techniques, as I'll describe. And basically, it allows you to count individual binding events between proteins and RNA molecules. And so this is how this technique works, at least the version that as we've implemented it in our lab. So what we do is to feed cells with thiol-modified nucleotides. Important to note that this does not have adverse effects on the cell, at least not for a day or so. And under the conditions that I'll be describing to you, the cells that are fed with these ribonucleotides produce essentially undisturbed titers of infectious HIV-1 particles. What one then does is to give the cells a zap of UV light, and any RNA binding protein that's in contact with a thiol-modified nucleotide will be cross-linked to it at some frequency. And so at the point at which the interactions are frozen in place, the cell is actually alive until you give it the shot of UV. Then what one does is to mash up the cells, treat them with ribonucleases, either RNase A or T1, to digest to the RNA molecules to a size that can be nicely mapped with high resolution to a site either on a viral or a host cell genome. Then the RNA binding proteins are immunoprecipitated with as much purity as one can muster. And we've actually spent quite a long time optimizing this technique. And everything I'm going to tell you about today involves the HIV-1 Gag protein, where we've introduced 3 copies of an HA tag in the flexible region between matrix and capsid. And we've also developed a polyclonal NC antibody, which we can use both for immature and mature virions. This tag is obviously mostly useful for immature virions. And so this shows that we're able to get almost pure Gag protein from these immunoprecipitations. The NC antibody performs almost as well. And so what you then do is to take your immunoprecipitate, do a couple of enzymatic steps to prepare the oligonucleotides for ligation and labeling so that you can see them when you fractionate proteins on a gel, cut out the species that one is interested in, digest away the protein, adapt to linkers, and then send it off, well, ligate linkers and then perform Illumina sequencing. An interesting feature of this particular approach is that the point of cross-linking often induces reverse transcriptase to make an error during reverse transcription, and so the actual nucleotide that is cross-linked to the protein that you're interested in is marked at a given frequency by a mutation. And so I'm going to tell you some results of HIV-1 Gag binding to the viral genome. Seb Lekutluay, who's done all the work on this that I'm going to talk to you about, will tomorrow present a second part to this story, which is arguably, actually not arguably, is more interesting than the stuff that you'll hear from me tonight. So this is essentially the footprint of HIV-1 Gag binding to the viral genome in cells. And so the way this data is presented is as a read density, and basically that is the frequency with which each nucleotide on the HIV genome appears in a read that's pulled out with the HIV-1 Gag protein. And there are some things that are expected, and there are some things that are a surprise. So we're reassured to see that the Gag protein binds to the 5' end of the genome where the packaging sequence is. Perhaps a bit more surprising is that we find Gag frequently bound to the Rev response element. There's also some other sites of apparently frequent interaction near the Nef start codon and again towards the 3' end of the genome. Now when we do this with a different strain of HIV, obviously things are variable here, so it's nice to know that what you're looking at isn't an artifact of the prototype strain you're using. Obviously the Psi sequences, the Psi interaction is conserved. The interaction with the Rev response element is also conserved, but these other things down the 3' end are a little less conserved and perhaps then therefore of less interest. And so I'll just show you a little bit of detail just to show you what the technique is capable of. So this is HIV-1 Gag binding to the Psi element. Of course, we expected HIV-1 to bind to the Psi element, but this gives a level of detail in the interaction that I don't think has been fully appreciated before, and certainly not demonstrated in the environment of a live cell. And so looking at the 5' end of the HIV-1 genome, we see essentially 3 peaks of interaction. This 4th peak here may be real, but it's difficult to assign significance to it because it occurs in the R region. And of course, there are 2 copies of R in the viral genome, so you don't know if a read in this region comes from the 5' end or the 3' end. So this is very likely an overestimate of how frequently the Gag protein is bound at the 5' end of the genome. But you can see that these interactions are quite spaced out quite far in linear sequence, but if you, at least in 2 dimensions, fold up the 5' end of the viral genome into a form that it is purported to adopt, then you can see that the peaks or putative sites of interaction with the viral RNA are quite closely spaced. So the binding to the RRE is very interesting. I very much wish that I could tell you what the function of this apparent interaction is between Gag and the HIV genome. I simply can't. We're obviously aware that there's a literature implicating the RRE in viral RNA packaging, but despite our best efforts, we have been unable to clearly show that RRE plays any role in packaging. We could be wrong about that, but we at least are immune to the charge of wishful thinking in that negative result. But anyway, the Gag protein doesn't bind to the same place as Rev, which primarily interacts in this position. Rather, at least with NL43, our primary sites of cross-linking are here. That's preserved in NDK, but we also get the opposite side of the stem in its interaction with RRE. So one thing that CLIP is useful for is when you think about HIV-1 Gag, its form and the way it potentially interacts with viral RNA could change depending on where you get the Gag protein from. So in fact, it looks as if the interaction between Gag and RNA changes utterly in a virion as compared to a cell. So here's the binding profile I showed you for a clip performed on cells, and this is the profile in virions. And you can see there's essentially no relationship. Instead of these rather apparently specific binding sites, the binding profile across the genome in virions has a somewhat disturbing appearance of white noise. However, this profile is very reproducible. In multiple experiments, the peaks and valleys are absolutely, almost, well, not perfectly would be putting it too strongly, very, very well conserved, and they're quite robust to changes in the methodology. So if we use a different cross-linking ribonucleotide, if we use a different ribonuclease to cut up the immunoprecipitate, if we use a different antibody to immunoprecipitate the Gag protein, these peaks and valleys are at least for the most part, conserved, suggesting the possibility that there is something of interest underlying them. However, if you try to correlate them with nucleotide composition, with secondary structure as determined by shape, there's essentially no correlation with anything that we've been able to determine so far. Now, how does this get to this? Well, let's first look in a little bit more detail in cells. And so we can fractionate cells into a cytosolic fraction and a membrane fraction by doing a membrane flotation reaction. And you can see the profile at the membrane is quite a nice hybrid of what one sees in the cytoplasm compared to what one sees in virions. And so you can still see the psi interaction and the RRE interaction to some extent. So clearly there's some, these aren't complete virions, but that signal changes to as I've mentioned, a completely different signal as virions are assembled and released. There's yet another change when virions mature. So everything I've told you so far has been done with an intact Gag protein, so in the absence of a viral protease. If you do the same experiment in the presence of an active viral protease and then use a nuclear capsid antibody to either pull down an intact Gag protein from protease minus virions or NC itself from protease plus virions, you can see that some of these specific or more localized interactions rise to the surface when NC is liberated from the remainder of the Gag protein. And so that leads to a model or a summary of what we've been able to find with these clip experiments. And Sebla will elaborate on this tomorrow. And so what we envisage is in the cytoplasm there are rather discrete sites of interaction between Gag and the viral genome. As virion assembly is initiated on the plasma membrane, those sites become less discrete, and then as you proceed through assembly, there are very, very many sites on the viral genome that appear to be in direct contact with Gag. And as virions mature, then the more discrete binding rises to the fore, although there clearly is interaction between NC at many sites along the viral genome, just less frequently than in immature particles. And I guess one could think of this in terms of NC concentration. And so you can imagine that in a lattice like this, the sort of local concentration of nuclear capsid is very much higher than it would be in this situation, or indeed in this situation. And so these differences might really reflect differences in the sort of the Gag or NC concentration with respect to the viral genome. And so that is more or less everything I wanted to talk to you about science. I'm going to go a little bit off topic for a few minutes to talk about something personal. And so a couple of years ago, well, actually, let me go back a little bit further and back to the time when an extraordinary run of good luck appeared to happen to me and my family. First, our first child, Stefan, was born more or less simultaneously with the discovery of tethering, which was, as I've shown you, elicited feelings of great euphoria. As I say, this marked an extraordinary run of good luck. The following year, I became a Hughes investigator. Then I had a second child, and then I got tenure at Rockefeller, which was a big deal. Rockefeller is one of the few places that HHMI investigators have been known to be denied tenure on occasion. So, and I got an award, and everything was going just fine. In fact, we were very secure and happy and immune to any apparent problems. But then the wheels fell off. In April 2012, I was diagnosed with stage 3 colorectal cancer. A few weeks later, my wife, Theodora, was diagnosed with stage 1 renal cancer. Basically, our lives changed within a space of a few weeks. Fortunately, Theodora's condition was straightforwardly treated, although it was very debilitating and very painful surgery and recovery from surgery, and she was recovering while I was being treated for this condition. My situation was, in principle, a little more serious and involved a rather debilitating series of treatments which I can only liken to having a truck thrown at you. So first I was poisoned for 4 months with this set of drugs, and then I was both poisoned and fried with drugs and radiation. I didn't feel very well after this. A low point in this whole scenario was throwing up on the street outside Rockefeller University. Then a brief break, and then the fun really starts with a surgery in which the last foot of my colon was removed, another brief period of recovery before a second surgery. So going through all this gets you to this point. Now, this was obviously quite distressing, and I didn't think about retroviruses for the best part of a year. Instead, I was reading papers by people such as this. It was actually quite reassuring to me that a significant part of the literature on this disease was written by the people who were treating me, a rather remarkable set of physicians at Memorial Sloan Kettering Cancer Center right across the street from Rockefeller. So as I say, after going through all this, you start at this point. And which one of these Kaplan-Meier curves you follow really depends on how all this went. So if the tumor wasn't very well controlled by the treatment, then things don't look that good for you. And more than half of these people will succumb to recurrence of their disease, which has a whole other set of very much more depressing Kaplan-Meier curves associated with it. If, however, you're lucky enough to get a complete response, then your Kaplan-Meier curve is up here. And I was extraordinarily relieved, and I'm not ashamed to admit I broke down when I got the pathology report from my surgery, which told me that my Kaplan-Meier curve would be this one. So I tell you this not just to evoke expressions of sympathy from you, although that's nice. But I sort of feel a responsibility to deliver a public health message that's actually reasonably important, because this is a very common disease. It's not one that's talked about very much, but there are 132,000 cases and 52,000 deaths in the United States alone each year. If I can just ask you to notice that you are sitting on a row of about 20 or 30 people, 1 or 2 of you in each row are going to get this disease. Okay? The good news is that most cases are preventable by colonoscopy screening. Almost all colorectal tumors start their life as a polyp, and they are observed during colonoscopy and removed right there and then, and you'll never know that you would have had the disease had you not been screening. So I implore you to look after yourself and go for this type of screening. There are various other common cancers which are also easily screened against, which I won't go into in the interest of time, but trust me, colonoscopy is not that pleasant, but it's much better than what was on the previous slide. Trust me, I've done both. Okay. So let me wrap up by thanking some of my colleagues, particularly those that have contributed to the work. So Sebler, as I mentioned, did most or all of the clip work that I told you about. I want to mention in particular Trinity Zhang, who's my lab manager. Generally doesn't get to come to meetings like this, but really helps the lab run and has contributed to very many of the projects that I've told you about. Melissa's work on MX2 has been presented at this meeting. You're going to hear from Rachel tomorrow. Matthew did the work on, some of the work on the species-specific aspects of VPU and tethering that I told you about. Alumni of the lab are listed here. I particularly want to mention Sam Wilson and, where is she, Shalini Yadav. I can't find her, you find her. Both of who contributed to the discovery of MX2, at least our version of the discovery. Mike Malin and Chang Lan's lab had an equally, arguably more important role to play in that discovery. This is Juan Martin Serrano, a former member of my lab. In fact, these are 3 of the first 4 postdocs that I hired, and you can see them using their combined massive intellect to work out how to use the remote control on this camera that Juan just bought. This is David Perez Caballero, who also has been was a long-time member and great contributor to the lab. My lab's a very joyful place, spontaneous expressions of delight at bands on gels. Apparently we don't give acting lessons at ADA. We've always very much enjoyed coming to this meeting. Here's some more people of the lab, in the lab. This is Stuart Neal, who many of you know, who was mostly responsible for the discovery of tethering. And this is Nolwenn Juvenet, who did the imaging work. I've been lucky to work with a very large number of great collaborators. I'm not going to go through them all, but I'd just particularly mention Vineet Kawaramani and Jeff Lifson's group, with whom we do the Simian tropic HIV work. Sandy Simon, with who I've done the imaging, and Charlie Rice, with who we've done lots of ISG screening to try and find new restriction factors. And we're doing lots of clip work with our center led by Alice Telesnitsky to study the interaction between various RNA binding proteins and the HIV-1 genome. Finally, I'd like to thank all of you. I've been coming to this meeting for a number of years now. I've made very many good colleagues, acquaintances, and friends. I'd particularly like to acknowledge the role that the veteran or senior members of this community play in their mentorship that generally doesn't get well recognized by the rest of us. The sort of diffusion of wisdom from them is something that we should recognize and appreciate. I was honored to help organize this meeting in 2005, and apparently this propensity to wear silly grins when one is here is something that extends all the way back. Apparently, though, I'm not the only one who demonstrates this phenotype. Although the underlying cause, while implied here, is rather more obvious here. John, of course, remains his normal distinguished self. Or perhaps it's just too early in the evening. The other symptom of being at this meeting is that sometimes it makes you think you can dance. Here's Alan Ryan and Jonathan Stoy showing us some moves. So hang around tomorrow, maybe you'll get some tips. But the only piece of advice I'd give to young investigators at this meeting is the meeting doesn't really start until after the bar closes. And so instead of these rather perfunctory exclamations of nice talk, if you really want to know what people thought about your talk, hang around here at about midnight, where these gatekeepers of scientific rigor will tell you how you really did. Finally, I'd just like to show you some pictures of my children for no apparent reason. Other than they're particularly cute, at least in my view. This is Eve, and I think she is going to go on to realize my unfulfilled ambition of being a rock star, judging by my inability to stop her from singing at inopportune moments. But you should probably keep your eye on this chap. This is my son, Stefan. He's 6 years old, and this is the sort of thing he draws for fun. And I hope you'll notice that the base pairing is correct. Finally, and furthest from least, I would like to acknowledge the role that my wife, Theodora, has played both in my career and my life. As I told you, we've been through a lot together, but we're partners in science, in life, in parenthood, and in everything. And she has made me both a better scientist and a better person. So for that, I thank you. And I thank all of you. Thanks. Thank you.
I couldn't let them stand up, huh? I'm speechless. That was fantastic. Questions for Paul?
Wonderful. I really appreciate the power of the par clip technique you were talking about, and I am not an expert in assembly, but it sounded to me like when I listened to Alan Ryan that RNA plays a big role in causing Gag to assemble. So I'm wondering, when you did your sequencing, do you see other RNA species bound to Gag at the earliest stages of assembly?
Yes, and you need to come to Sebla's talk tomorrow to hear about that. I will not steal her thunder.
What else? Who else has a question? There we go. Thank you.
So Paul, how about Appleback 3G binding to RNA?
Are you going to talk about that tomorrow? No, okay. Then fine. Whoops. Oh no, we've lost it. It's all right, get it back. So these are preliminary data. Basically, it's everywhere. So in cells, you see a sort of a pattern of binding where you get enrichment at the 5' end and then gradually towards the 3' end. This is sort of what you'd expect if it was bound at many places, more or less equally represented across the genome, because of course recognize that you have completely spliced RNA, which will be represented here and here, partly spliced RNA that will be represented here, and then full-length genome that will be represented here. In virions, again, many sites across the viral genome, but absolutely no correlation with Gag binding sites on the viral genome. You probably want to know about 7SL as well. Let's talk about it afterwards.
Any other questions for Paul? There's one.
Very interesting talk, thanks for that. Is there any reason that your read density in cells is much greater than in virion, and could that be related to this pattern that you see?
No, that's not a biological difference. So when we send the things for, actually the signal in virions is significantly stronger than the signal in cells. When you send these things for sequencing, you put more than one experiment in each lane, and so your number of reads that you get is determined as much by the number of experiments you load in one lane as it is by the any biological difference between lanes. So we always try to get as many reads as we can. You can see with APOBEC the numbers aren't quite as high as we'd like. That's because there just isn't as much of the protein in virions as there is as GAG protein is in virions. So basically, no, don't attach any significance to those numbers. You can compare across a sequence, but not really effectively between experiments.
Can you tell how many molecules of GAG are for any genome?
No. No, these are essentially unlinked reads. This is a frequency distribution of how often each sequence is bound to GAG. What the signal here doesn't give you any information. If you have a signal on an RNA molecule here, it doesn't give you any information about here. This could be 1 million different RNA molecules, each one individually bound to a different APOBEC protein. Or it could be one RNA molecule with millions of APOBEC proteins. Obviously, that's not how it is, but those two extremes are theoretically interpretations of this type of data.
Paul, is there any way to get the reciprocal, well, not reciprocal, any information about which part of GAG is binding to these points?
Yes. Come to Sebla's talk tomorrow.
Anyone else? All right, then let's have another hand for a terrific talk.